Howdy. So, this is a good thing. Normally this is when I arrive and then I'm sort of madly plugging things in, but we're ready to go. For those who don't know, I am not Dr. Alvarado. I'm Mitchell Baker. You can tell us apart because he's cleverer. I am older and smaller. That's about it. So I just want to go over a few things. One, a couple students asked if they could record lectures. So if you're here, and especially if English is your first language, that seems like an awful thing. But you should also know that I record my lectures and I will upload them in the Google Drive, the videos, which is going to have my lectures. I thought actually there was another folder that says lecture recordings. There probably will be. But anyway, the Google Drive for this class, where you hopefully were able to sign up for species things. Now you really want to have access to that folder, perhaps more importantly, because I'm going to upload recordings of my lectures and sometimes other large files or videos that I might want you to see. So next week there's a midterm. It's on the first 10, which means the first 5 lectures, so it's not on tonight's lecture. Don't worry, tonight's lecture will be on a midterm, just not next week's. In order to give us more room to spread out, we're actually going to split the section into 2 rooms. So if your last name ends in A to J, meet in Kylie 150. We'll have a couple proctors there. And if your last name is K to Z, meet here. We've never actually had both large rooms, so you'll really be able to spread out. It's a nice thing. Bring your ID, because most of the time it doesn't matter, but every once in a while someone does something really bizarre. And so especially if your TA isn't in the room and recognizing you, if that's the case, then don't worry about your ID. But bring your ID to hand in the test. As you know, and you've gotten from the announcements, just as a few pointers, different professors teach differently, and your job, unfortunately, is to quickly figure out what our styles are. I hand out these sort of hard study questions that you're supposed to break your teeth on and work with in groups and stuff. And then the exam questions are versions of those multiple choice versions of those questions. And Professor Alvarado is not doing study questions, but he uploaded links to all the Kahoots from lecture. I looked through them. They're pretty straightforward. You should absolutely go through all of the Kahoots that are uploaded. And also notice, the clear things about lecture sort of style and stuff. Dr. Alvarado is using the lecture slides that are associated with the textbook. So there's a pretty tight link. So if I were you, I would work from the slides mostly, use the textbook, and even your lab manual for the diversity stuff like prokaryotes and protists, but focus on the slides. And that would be my advice. Yeah? My lecture? So, as I said 5 minutes ago, sure you can. There's one student's phone right here. I am recording this lecture now, and I will upload that recording. But, maybe my computer explodes and, whatever. The ones that I upload are going to be fairly large files, about 500 meg, and you want to download them because it's a video. It's like me and anything that I write on the screen while we're lecturing, which is somewhat important. You'll notice for me, it's useful, I think, to print out my slides before a lecture if you hate trees and want to do really well. Print them out 4 slides per page and double-side print. Don't kill more trees than you have to. But be ready to write on the slides because my slides are not complete, right? I will often leave the most important thing as something that I say and maybe something that I write on the slide. It's not going to be on the slide before lecture. And then the last thing is that I will also be using Kahoots. That's a new thing. I used to try doing Blackboard quizzes in lecture. They're graded just on participation. So I'm not totally sure how Professor Alvarado did it. When I call it up, I'm going to ask you to enter your CUNY ID as your nickname. And if everyone does that well and carefully, then I will have a record of everyone that took the quiz, and I'll also, have all the good stuff from those quizzes, like instant feedback, no one understood that, or, good, everyone got that. So there you go. Okay, so tonight's lectures are speciation and how to use phylogenies. Oddly enough, they're kind of reversed in order in the textbook. So we're first going to do chapter 23, then we're going to do 22. Not 100% sure why they're ordered like that in the book, but there you go. So the first thing about species is that people argue about how they're defined. And you'll notice in the text they mention 3 different definitions that can be useful in different contexts. The text talks about morphological species, do things look the same? And that was the original species concept. But as we've learned now, sometimes organisms in the same population can look really differently, but they can mate with each other and they share genes really freely. Sometimes they look really similar, but they manage to avoid mating with each other. We'll go over those. That has to do with prezygotic isolating mechanisms, like they mate different times of the year, or they have different songs that only some like and others don't. So that's the problem with the morphological species. Another definition of species that I'm not going to go into quite as much, it's in the text, is the lineage concept. The idea that, okay, maybe there's some gene flow, but if a lineage can kind of evolve independently of another lineage and respond to environmental differences adaptively, it's a species. And that's actually probably the best definition, but it's fuzzy. So this is an intro course, so we'll mostly focus on the biological species concept. Biological species concept, it's what's up there. Biological species are all the individuals capable of producing fertile offspring. That's the species. It's kind of like that joke about democracy. Democracy is terrible except for all the alternatives. Biological species concept has a number of flaws. We'll go over it, but it's the least subjective. It's the most objective, easiest to sort of measure, define clearly. If you could potentially mate with another individual, you're in the same species, and produce fertile offspring, you're the same species. Okay, so if reproductive isolation is what defines species, that's the biological species concept, then we need to understand what maintains reproductive isolation. So this is going to be the prezygotic and postzygotic barriers. Prezygotic barriers, these are things that block mating before fertilization could occur. Just the definition of the term. One example is geographic isolation, and that isolation could be due to distance. Two populations could be separated by such a long distance that you're never going to have gene flow between them. But it could be a barrier like the Grand Canyon, which looks like a pretty permanent thing, but actually it's not. It's a few million years old. You have a river, you have sedimentary rock. You give it enough time, you can get a canyon. And so this has happened, this has created a barrier that has separated many populations, including ground squirrels. So there are ground squirrels on either side of the Grand Canyon that now have been separated for a few million years and are at least on the way to becoming separate species. So if you can have a physical, geographical barrier, that can aid speciation. Prezygotic barriers also include behaviors. I've sort of hinted at that before. And those behaviors could include habitat preference, and as I'll talk about later with the apple maggot fly, habitat could mean something like what fruit do you settle on to look for mates? It could be something really subtle, like crab apples versus regular apples versus cherries. Or it could be, a habitat that you would recognize as more different, like different altitudes in a mountain. It could be temporal variation. What season are you breeding? Again, that's going to come up in the apple maggot flies. And it could be what kinds of individuals do you like to mate with? And this is where you can see some of those examples of cryptic species. In the text, there was an example of two species of tree frog that look really, really similar to each other, but they're behaviorally separated, and we'll talk about some of the frog examples in a second. But one example that's not in the textbook are crossbills. So Loxia curvirostra, I think it's still officially one species, but like 30 years ago, there were some really good studies showing that actually there are 7 different subspecies. It's hard to say exactly what how they're classified, that are at least partly reproductively isolated. So crossbills are these really cool adapted birds. They're called crossbills because their bill doesn't meet in the center. It's offset each bill so that they join like this, and that gives them the ability to wedge open the blades of pine cones and then get at the seeds inside. However, really good ornithologists who are really sensitive to basically bird calls notice that there are consistent differences in the calls that crossbills were giving when they were feeding on different species of pine tree. And it turns out that, there are 7 different subspecies that by and large specialize on different species of pine trees. And actually they fly to different parts of the country depending on where their favorite species is producing the largest crop of seeds. So it's really cool. I have a link to where you can hear the different calls. They're often pretty subtle. For some reason, I wasn't really able to get these guys to play. Let's see, is it gonna? It's kind of subtle. Let's see. Okay, sometimes it works. Okay, so that's a flight call, but I can't get this guy. This guy's flight call is not going to fly. I don't know why. And this is an alarm call. The alarm call is going to be actually a little subtler and quieter than the flight call. But in any event, you can see in the sonograms that subtype 2 is different from subtype 1 in each of these calls. Sadly, we can't hear them. But if you go to this website, if you go to eBird rec type, it will give you the link to their different calls. So their geographic range overlaps, but they have a prezygotic isolating mechanism. They have two, really. One, that they choose different habitats. They choose different pine trees to specialize on. And they have differences in calls that affect mating preference. Another thing, so there's no strong reason for you to know, I'm mostly an entomologist. And a lot of the insects that I study often have cryptic species. And one of the things that will distinguish species that basically look the same, like a whole bunch of fruit flies that all look alike, but if you examine their genitalia on a microscope, and this is really hard, not just because they're small, but because the genitalia are inflatable, and so you try to dissect them out in a microscope, and all you see is kind of like a sack or mush, but when you can get them inflated, you'll see that they have very different shapes. Females, very different inside. Males are very different in their genitalic anatomy. And the idea is that actually this is part of like an coevolutionary race between males and females, and possibly selected to avoid mating with the wrong species. So you have these really complex female genitalia and equally complex male genitalia that will only match the right species. So there you go. That's another example of a prezygotic barrier. Postzygotic mechanisms, after fertilization occurs, the offspring dies or the offspring develops but is infertile. And so the classic example of that is the mule. A horse and a donkey can mate and produce a physically strong and healthy offspring, a mule, desirable in some contexts, good for work, but that mule is not going to can't produce offspring. So that maintains them as good species. Another example of postzygotic reproductive isolation are eastern and western meadowlarks. So this is it's related to an example in the text of several species pairs of fish that became geographically isolated during the most recent ice age. So glaciers spread down through the Midwest, separating what used to be sort of continuous wetlands into separated areas. And in the text they talk about different, 4 different pairs of species that have evolved because of the most recent glaciation. That happened in birds a whole bunch of times as well. So there used to be one meadowlark, but sometime from the last glaciation, or maybe the one before that, 40,000 years ago, there was geographical isolation. Now there's an eastern and western meadowlark, but the glaciers receded, and humans have changed the landscape and made dispersal easier in some ways. So a lot of these populations or species are now coming into contact. In the meadowlark case, they produce hybrid offspring just like a horse and a donkey, but the offspring are sterile. And so genes from one species don't get mixed with the other species. And what you expect in this situation is that over time, these species should evolve prezygotic isolation as well, right? Because it's a waste if you mate for a season and feed the offspring and then they don't have any offspring themselves. So that should select for isolating mechanisms. Yeah?
So two species are genetically similar, but similar enough, then they would be able to produce a functional offspring.
Functional is a fuzzy term. So if they produce fertile offspring that then can continue to mate and reproduce, then you'd say they're the same species. If they produce healthy offspring, but offspring that can't reproduce, you would say, no, they're separate species. They're two different species. Yeah?
But two species, they're different enough to be considered different species, but they produce offspring that could reproduce.
Yeah, so that happens, right? And that's the problem. One of the problems with the biological species concept is sometimes you have what seems pretty different, differentiated populations or subspecies, and they can produce viable and fertile offspring. We'll talk about that with the Rhagoletis pominella, the apple maggot fly. And basically, biologists argue. So Ernst Mayr, like one of the most influential evolutionary biologists, he died a few years ago, he was 100, he would have said, if they can produce fertile offspring, it's the same species, it's not different species. Other biologists who are more interested in like variation within species would argue that they are still species because they're evolving in different directions, and like there's disagreement. So people would argue about it. Yeah.
So in the previous slide it said something about having grandchildren.
Yeah.
Yeah. So does that mean that they can have viable offspring?
No. I'm saying the parents hybridize, they produce offspring, but the offspring are infertile, so they can't produce grandchildren for their own parents. Right? Yeah. Okay, so we're bumping up against some of the limits of the biological species concept. One I hadn't mentioned at all, like you had a whole lecture on prokaryotes and protists, many of which are not sexual, right? They just reproduce by binary fission. And then there are even multicellular organisms that don't have different sexes. And does that mean none of them have species? Are there no species of bacteria? So biological species concept just doesn't apply to some classes of organisms, including asexual species. Biological species concept also is not testable for fossils, right? And that's also something to keep in mind when we're looking at fossils. Our definition of species for extinct animals is different than the ones for living animals. Like, you probably can't tell like something that's as different as a lion and a tiger from a fossil, from fossil remains, even though you can from looking at living organisms. The other thing is, let's say the question isn't testable because the populations are geographically far apart, so they never get a chance to mate. But if you could put them together, they would mate. What do you call them? So sometimes that's a practical issue. If you have the budget, you might be able to test them, but sometimes you can't mate them in captivity. And then, your question was, what if we think of them as good species, but they can happen to produce viable hybrids, and then so you get like the Rhagoletis pominella example is a good one. They totally can produce viable offspring, and there's like 5% gene flow between these two species or subspecies every year, but they also have consistent, strong genetic differences in things like what fruit do they like to lay eggs on, and what's the timing of when they hibernate and when they mate. And so people argue about whether they're good species or not. I sort of lean up to they are species even though they violate biological species. Yeah.
So when two species, like the mule, cannot have offspring,
Right.
the mule still considered a species?
No, it's considered like a hybrid of two species, and it's kind of nothing. I mean, as an individual, it might have a full life, but yeah.
What do you mean by a good species?
Like a well-defined species, like clearly a species separate from another, as opposed to some of the examples that we talk about, where it's like maybe they're the same species, maybe we're talking about two different species. And so good, I would define as able to evolve independently from another species. Yeah.
Is it possible to genetically engineer one species to be actually similar enough to another species and create an offspring that is fertile?
Oh, that's a good question. So I think the opposite is much easier, to like insert genes or to create chromosomal mismatches to instantly create new species. The opposite, harder. Possibly if the genetic mechanism of isolation is known. Not totally, actually so it's a good question. I don't think anyone's ever going to bother doing that because probably the parsimonious approach is if you want to get the traits of one species into another species, we're now like targeted enough that you just find that gene and like, you know, use plasmids to get it into yeast and then or whatever your vector is and insert it into a genome and just do it trait by trait. I don't think anyone is now trying to study a way to like allow different species to hybridize because like a more targeted approach is going to be cool. So these other definitions, like in the text, morphological species, genealogical or lineage, that's what the textbook had. It didn't say genealogical, but that's what it means. Let's see if we can do this. Nope. Come on. In the text, they said lineage. Those other definitions apply because the biological species concept, you know, breaks down, can't be used in fossils, can't be used for asexuals. Okay, so the next thing is like, what makes it easy for new species to arise? And the answer is, cool. I'm going to skip to the take-home. It's going to be much easier in allopatry. But let's get to that. The first thing is definition. What does allopatry mean? Different. Close. So allo totally means different. And patry, geographic location. So, you know, I'm not even sure, like, it might have relations like with father, like fatherland, and you go from father to fatherland. But in any event, the patry part is different location. And so that means that sympatry means same location. And, you know, like I said before, geographic barriers, geographic reproductive isolating barriers will make it easier for species to arise. And that's, you know, the concept we're gonna develop, especially with like Galapagos Islands and fruit flies in Hawaii examples. That's what's coming up. So it's much easier, it's much more common for new species to arise when they get divided, when they get split up into separate areas in allopatry. And that's for a couple reasons. One, allopatry shelters genetic variation from gene flow. In other words, when a species is separated in two areas, because they're separated, there's not going to be genes passing between them that's going to make them homogeneous, that's going to make them the same. And then either selection or genetic drift can then lead to reproductive isolation. And you should read the text for that. It's good. It talks about some different interesting things. It talks about the Muller and the other guy mechanism that was proposed. Like, it was a really good question. You know, text talks about it well. It's like, hey, if sorry, what? 23, I think. It's definitely Muller. Miller and Urey is like generating life chemically, amino acids. No, Muller, M-U-L-L-E-R, famous population geneticist. Muller's ratchet is the other thing that he's famous for, but it was Muller and someone else. The question is like, a gene for reproductive isolation should be terrible. It arises in a population and now you can't mate with anyone else. How could you ever have different species evolve? And that's true. But the idea is that in allopatry, you can have one mutation that doesn't lead to reproductive isolation, but it drifts to fixation. In the other population, another mutation in another locus also doesn't lead to reproductive isolation, but it goes to fixation. Now, the combination of these two new mutations leads to reproductive isolation between them. So you can have, in allopatry, you can have just a whole bunch of genetic changes happening, none of which individually lead to reproductive isolation, but then when you bring individuals from these two separate areas together, now they can't reproduce. And like a classic like natural experiment in speciation is the Galapagos. It's the system of fairly young islands, and they're separated by, tens or hundreds of miles. Not that far for a bird to fly, but far enough that they don't fly in between islands every year. And there are, as we know, this like classic example of an adaptive radiation found on these islands. So there is a seed-eating finch that is, that lives in Ecuador and is the sort of closest relative to all of the Darwin's finches in the archipelago. And from this single ancestor, 13 species have been described. And not all of these species are found on all the islands. Usually, actually, each island only has a few. They've diversified in what they eat and their bill morphology and body size in a whole bunch of different ways. Actually, a lot of them look pretty similar, but their bills and their behaviors separate them. So, for example, you have from an ancestral seed eater, you have one species that specializes on flower buds. It just kind of chomps down on the flower buds and eats those. And then you have a bunch that eat insects. So even though the ancestor, specialized in like grinding, opening up seeds, you have one, and they eat insects in different ways. So you have like the warbler finch that has a slim bill that is like a warbler or a robin, which are small insect-eating birds that we have. You also have a highly specialized species, the woodpecker finch, that I'm going to talk about in a couple different ways. It smashes into both like mangroves and cactus and eats the insect larvae that are found inside the plant or under the bark. You also have seed eaters, but you have a diversity of seed eaters, some that have larger bills and smaller bills, some that specialize on large seeds or smaller seeds. Yeah, the niches are amazing. So the really cool thing about the woodpecker finch is that it is a classic example of convergent evolution with true woodpeckers. But this niche is a new one for this species. So North American, actually Eurasian, all true woodpeckers, they're not in passeriformes. They've got a bunch of adaptations. They have a super strong tail for holding on. They have 2 toes facing upward, 2 toes facing downward to grab onto trunks really well. And they have a super long tongue that sort of is coiled in the skull, and they can use that tongue to reach in and sort of spear and catch grubs, insect larvae, after they've smashed a little hole in the bark or the outside of the tree. The woodpecker finch smashes holes, but it doesn't have that tongue. It just hasn't evolved that adaptation. However, behaviorally, it has adapted to collect cactus spines and use the cactus spine as a tool to reach into the holes that it's smashed and impale and pull out insect larvae. So behaviorally, it's occupying the same niche, even though it's come into this, much more recently. It's not morphologically adapted to it as much. So how has allopatry helped in speciation? Well, what seems to have happened in this group is that you have the initial colonization from Ecuador, and you have a finch that settled in one island, probably the closest one, and then it has expanded its range into other islands. And at each new island, it's geographically isolated from the other population, and it diversifies in this new habitat, perhaps specializes on a different size seed or switches to eating insects. And with each new colonization, it's free to diversify more because it's not exchanging genes with either of these populations. And what happens is, at some point, that species recolonizes the descendant population, recolonizes the first island. And if it's different enough, these two populations aren't able to breed with each other, or they don't want to breed with each other anymore, and now we have two species. And so this process of colonization and diversification, differentiation, has happened a number of times. So now we have about 13 species on this group of islands. And you can see the importance of allopatry with kind of a natural experiment. So Cocos Island is another Pacific island. It is far more isolated than the Galapagos, and it is a single island. It's about 600 kilometers northeast of the Galapagos, and there's nothing else nearby. And Cocos Island also has a finch that's related to a mainland finch, but it just has one species. Whereas most of the Galapagos Islands will have 3 or 4 different species. Why? Because there's no allopatry. There was no opportunity for the Cocos Island finch to colonize another island, become different, recolonize, and now they're 2 species. So without that subdivision, without allopatry, it hasn't been able to diversify into many different species and occupy many different niches. So that's kind of cool. So allopatry, definitely important. However, allopatry doesn't always lead to new species. When a descendant population recolonizes an ancestral population, sometimes they fuse, like they're not isolated, and they merge again. Sometimes populations that come in contact after being separated for a while form a hybrid zone. That hybrid zone might be caused by geographic barriers or might be caused by partial reproductive isolation. Sometimes one species just takes over and wins. So I'm going to go over some of these. There are examples of each. Sometimes they come together, and like some of the Darwin's finches, they're good new species. And sometimes we have this process called reinforcement. So I'm going to talk about that now. Reinforcement is when subspecies or populations in sympatry become more different than those subspecies or populations in allopatry. I wonder if I wrote that down. I didn't, so I'll write it down here. Besides, it kind of slows things down, gives us a chance to think about it. Reinforcement. It's when subspecies or populations in sympatry are more different or become more different than the allopatric populations. Okay, so they're becoming more different in mating behavior, mate preference, in prezygotic isolating mechanisms. And why is that going to happen? That's going to happen if the hybrids are less fit. So like if the hybrids are sterile, that's an extreme example. But maybe the hybrids are just not quite as fertile, or the hybrids are not able to specialize in one adaptive niche or another. If the hybrids are less fit for any reason, that's going to now lead to selection, figuring out ways to avoid making hybrids. So there are a bunch of examples. This is one that's, I forget if it's in the text or if it's just really related to one in the text. There are a few of them. Green-eyed tree frog, Literia genimaculata. In this study, there's a northern population and a southern population. These are two, not sure if they're species, subspecies, people argue about it. But what's really interesting is at this area where they meet in Australia, there is an isolated region of southern green-eyed tree frog. How does that happen? The northern and the southern areas meet. There's actually like a river, but then the northern population expands its range into the southern and then creates this little isolated pocket. What happens is frogs well in the north or frogs well in the south never have a chance to meet a frog from the other population. And so there's no selection on them to avoid mating with the other subspecies. But ones right here in this isolated pocket of the northern population, the isolated southern population, it's a small subpopulation, and they are much more likely to run into individuals from the southern subpopulation. Hybrids are less fit, and so actually there's now selection just on this little northern subpopulation to avoid hybridizing. And what's happened is that individuals in that isolated southern population are now more different from the northern than ones that are, far within the southern population. So the prediction of reinforcement is that insulated populations not exposed to the other one, don't have any selection to avoid mating with the other one. But ones that are next to each other, they are brought in contact, their offspring might be less fit, and if that's happening, that will select against mating and make them, the ones in close contact, more different. And that's what they saw. So in this figure, and always you want to look, understand the axes whenever you see a graph. So in this case, you have 3 different categories of frog: northern, southern, and then isolated southern. Frogs collected from that little snippet island. And then here we have 2 dependent variables, body size and call, a bunch of variables in the call to describe the call combined into one. And what you can see is that, sure, the southern is a little different from the northern frogs. The southern are a little bit bigger on average, but the isolated southern are very different. And they're actually very different from either the regular southern or the northern. But they're more different from the northern than the southern. What's confusing about this example is when I say isolated southern, I mean it's that little island of southern surrounded by northern. So it's isolated from the rest of the southern population, but it's not isolated from the northern. It's actually really exposed to the northern population. Here, you can see in call, again, the southern has, on average, higher pitch. I think that's what this is talking about. But the isolated southern is more different from the northern than the rest of the southern. So this is an example of reinforcement, and this is an example of reinforcement. That in sympatry, in close contact, the subspecies have been selected to be more different. Reproductive isolation is evolving due to reinforcement, due to hybrids being less fit. Okay, this is the same story, but two different subspecies. This is the example from the text. And what you can see is that in allopatry, they have different call frequencies, and in sympatry, the difference is greater. So that's what's important about this figure. The difference in call frequency, that's how high or low the call is greater in sympatry, than in allopatry. It's a smaller difference when they're geographically isolated. Okay. Another example of reinforcement. This is another pair of frogs. So clearly frogs are interesting and have been studied for speciation a lot, which is true. What's important here is something a little bit different. So here on the x-axis we have the season of breeding. And in isolated allopatric populations, sometimes these different species, Rana is the genus, Berlandieri or Sphenocephala is the species name. So they're all in the same genus, but they're different species. And look, Berlandieri and Sphenocephala, they kind of breed, at different times in, February versus March, April. So Sphenocephala breeds more in early, late winter, and Berlandieri in more in spring. But then, look, they have a second breeding phase during the year in September, October, and they're breeding at the exact same time. But they're in allopatry, so it doesn't matter. They don't get a chance to hybridize. When they're found all in the same geographic location, there's a neat separation in the timing of breeding, right? These three species no longer overlap in breeding, and that's because they're in the same geographic location. So if they were singing and breeding at the same time, they'd be producing hybrids that would be less fit. Another example of reinforcement. Okay, so when two populations, maybe species that were separated, come in contact, they often form hybrid zones. And I'm not going to go into a lot of detail about this. You'll learn more about them in an evolutionary biology class like 287. I'll just say that population geneticists spend a lot of time analyzing these hybrid zones because they can tell you a lot about reproductive isolation and whether it's good or bad for these two subspecies to mate with each other or not. And these hybrid zones behave differently. Sometimes they stay stable for decades or even hundreds of years. Sometimes they form, and you can see on a scale of years that they're getting wider and wider. And what used to be, we thought were two species, are actually in the process of fusing. And you can see that in their bird and frog examples of all of this. So one is two bird species that were, again, like the meadowlark, separated in the last ice age, and they look pretty different, Audubon's warbler and myrtle warbler. There is a wide 150-kilometer hybrid zone in the Rocky Mountains where they interbreed, and this hybrid zone gets wider each year. So probably give them another 100 years, and you won't be able to say that there are two species, they'll have fused. Another classic example of adaptive radiation and the importance of allopatry is seen in Hawaiian Drosophila. They're kind of like the Darwin's finches, but crazy, but like much, much more. So there might be, depending on how you define species and how well they're described, maybe 1,000 species of fruit flies in the Hawaiian archipelago. And the story is the exact same as Darwin's finches, just flies have a much faster generation time, and for whatever reason, this cycle of colonizing, diversifying, recolonizing, becoming reproductively isolated, like it's much easier for reproductive isolation to evolve in flies. And so now there's hundreds of different Drosophila species on the different islands in the Galapagos. Another example are the cichlids in Malawi. This is an interesting example also because a lot of these species are now extinct because an introduced Nile pike has sort of wiped them all out. So they're like kept in captivity because collectors really love these different species, but a lot of them aren't there anymore. The interesting case about the Lake Malawi cichlids is that they have evolved multiple species all in the same lake. So it's really fascinating. These Rift Valley lakes are incredibly deep, very diverse habitats, different in how much light gets into different habitats, in whether there's sediment, sand, or rock. And the cichlid fish evolved many, many, many different species in a very short period of time. Okay, so the last 15, 20 slides was just to show that allopatric speciation happens much more common. What's much less common, but is interesting, is sympatric speciation. The formation of new species in the same geographic area without geographic barriers. Now this can happen in plants much more easily than in animals because plants have kind of a modular body, and they also can often self-fertilize, self-pollinate, and that allows for sort of instant species to form in plants in a way that it can't in animals, due to basically mistakes in gamete formation leading to changes in the number of chromosomes. In animals, that might be a dead end. In plants, because of self-fertilization and vegetative reproduction, you can sometimes actually get a new species due to these events. So one example is autopolyploidy, which is when you have hybridization within species. Basically, instead of forming haploid gametes, if you form diploid gametes by accident and they self-fertilize, you can have basically a doubling of the number of chromosomes that is now incompatible potentially with the parent, but is compatible with itself. So the simplest kind of meiotic error, producing diploid gametes, can lead to these new species with even number multiples of genes. So basically, instead of being diploid, they'll be tetraploid. And now you have a new species that can sort of evolve independently. And there are several sort of pretty clear examples of that happening in plants. Another thing that can happen is allopolyploidy, where you have different, you can have different species hybridizing, and you can have, so they have different numbers of chromosomes, they shouldn't be compatible, they produce an offspring that's not compatible with either parent species. However, because of vegetative reproduction, you can produce offspring that have the exact same weird new number of chromosomes. and those will then be sexually compatible. So you can have, because of vegetative reproduction, you can have basically an offspring that's compatible with neither parent, vegetatively produce offspring that will be compatible with each other. So also that happens in plants because plants have this modular structure and they can reproduce vegetatively as well as sexually. Okay, the last example that I want to talk about for sympatric speciation is in animals. And so you might have sympatric speciation if you have colonization of different ecological niches that can lead to at least some isolation between the two subpopulations or species. So this is controversial. Some population biologists think this is totally a thing, and some think it's not a thing. So I'll give you the best studied example, which is the recent speciation of Rhagoletis pominella, usually called the apple maggot fly, although it really should be called the hawthorn maggot fly, but now there's also a cherry maggot fly. So why do I say that? The native host is hawthorn. Apples, there's no strong reason for you to know, are not native to North America. They were, I think, first domesticated, cultivated in the Caucasus, like Kazakhstan. And then they spread, and they're an awesome fruit. This is the time of year to go apple picking. This weekend, you should totally do it. It's awesome. It's kind of expensive way to get apples, but it's so much fun. Anyway, there you go. The native hawthorn maggot fly made a host shift. In the 1860s, it's now being observed as a pest on apples. And actually then much more recently, in the 1960s, we're seeing the same species on cherries. And it's an annoying pest. No one likes to bite into this. And so it gets studied because farmers want to know how to control it. But here's the interesting thing. Apple trees and hawthorn trees ripen their fruits at different times of the year. So apple trees start their fruit earlier in the summer, in early July, whereas hawthorn trees have young fruit starting to develop in late July. And what that means is, what these flies do is they do their whole mating attraction thing, releasing odors, doing a little dance, laying their eggs on the really young fruit that are developing long before you want to eat them. And the young hatch and burrow into the fruit and eat on the inside, and they eat and they grow, and then at some point they eat the stem and cause it to weaken and make the fruit drop prematurely. And then they complete their development, they eat more of the fruit, and then they burrow into the soil to pupate, and they overwinter as pupae. So that overwintering stage, I want to mention it because it's just good vocabulary. You should know that overwintering stage is called diapause. Diapause it's hibernation for bugs, but it's a dormant phase that insects can go into. Interestingly, I'll talk about this a little bit more. Insects can diapause in any stage. Not any species, but you'll have different species are specialized at different stages. So for example, I'm going to do this again when we do arthropods. Webworms. It's a moth that that has colonies of caterpillars in trees. They overwinter as eggs. Woolly bears are a lepidopteran, a moth that overwinter as larvae. I study black swallowtails that overwinter as pupae, just like these flies, the Rhagoletis pominella. They diapause as pupae. Mourning cloak is a butterfly that overwinters that diapauses as an adult. So it's characteristic for each species, but they could do it at any stage. And so here's the thing. They usually the hawthorn ones mate on these later developing fruit. They go into diapause later in the fall. They have a shorter diapause. The apple, Rhagoletis pominella, they mate on the fruit earlier in the summer. They go into diapause in the late summer rather than the early fall. They have a much longer diapause. And the problem is that if these two subspecies, whatever they are, hybridize, they can totally mate with each other. Their offspring are fertile, but they diapause for the wrong length of time. When they emerge the next year, they are not going to be emerging in the right time of early or late July as adults to find mates on the right ripeness of fruit. So because they prefer different fruits and they've evolved a difference in their preference, they've also evolved a difference in their diapause time, and that restricts the flow of genes between them. So they are in the process of evolving reproductive isolation. It's not perfect. There's gene flow every year, but there are good genetic differences between these subpopulations. So it's a really interesting example of speciation kind of happening in real time. Okay. So this is kind of what I've said. They shifted host from hawthorn to apple. Now that changes the optimal time of mating and the duration of diapause. And they have diversified, they vary genetically, but there's some gene flow every year. Some of them pop out at the wrong time or find the wrong fruit. And what maintains the distinctness is that there's a trade-off. If you hybridize, you're going to diapause for the wrong time, and your offspring are not going to emerge at the best time for either of these, either fruit host species. Okay. Cool. So applefly pupae develop and gear up for a long winter earlier in the summer in warmer soil. Hawfly pupae don't go into diapause until the soil is quite a bit cooler, 22 degrees. They go into diapause usually in late September. So each one is specialized, and that selects against hybrids. Cool. To sort of end this, want to talk a little bit about why speciation is important. And the main reason why speciation is important, and the reason why population geneticists sort of care about it so much, is because once you have two species and there isn't gene flow, they can evolve independently. That's the meaning of this, highfalutin thing. Cladogenesis facilitates anagenesis. What does that mean? Cladogenesis, which means formation of new clades or splitting into species, facilitates anagenesis, change within a lineage. So this is kind of like the drawing version of it. Anagenesis is change within a lineage. You have this, it looks like a European roller that's changing color from dark blue to turquoise and green to red cheek patch. If you split into two species, that becomes much easier. Now you have two different groups. Nothing's going to smush them together. There's not going to be gene flow averaging them. And now this one's free to evolve red cheek patches and a crazy long tail and light blue. And this one, staying like the ancestral type. So splitting, cladogenesis, facilitates anagenesis, change within a lineage. And that, it's a basic concept, it's easy to understand, and it's true. If you have species that don't mate with each other, that restrict gene flow, now each of them are free to evolve in different directions, specialize in different niches. And you can really see this in the Galapagos finches, the Darwin's finches. So you can see this in different islands. In Daphne Island has only a single finch, Geospiza fortis. And that's the classic example that, like, in wet years, the smaller bill type is favored, and then when there's a drought, only the ones with the really large bills can crack the large seeds, and that leads to a big, quick change in the shape of the bills, and then it sort of bounces around. However, on other islands, there are other islands that have multiple species all eating seeds. So in Floriana, you have both Geospiza fortis and Geospiza fuliginosa, another species. Now that you have two Darwin's finches, fortis specializes as a large bill, I think, a small bill, and fuliginosa as a large bill. And now those two, because you have two species, you end up having them evolving more different bills than in any of the islands where you only have one species or another. In the islands where you have only one species, you end up with just sort of a generalist intermediate bill size. In the island where you have two species, they have specialized, and their bills have evolved to become more different. Yeah.
So they're the same species, but they just had modified their beaks over time.
So you got two species. On islands where you only have one or the other, they have a sort of intermediate bill size. On the island where Floriana, the island where you have both together, they're reproductively isolated and they're competing, and they've evolved to become more different so that each is now specialized in a different niche.
So if like a G4 is from Floriana and went to that, maybe they could still reproduce?
Yeah, exactly. If a fortis from Daphnia went to an island there where there was only where there was just one species, it could mate. It could mate with the original species. Yeah. So species are evolutionarily independent, and what that also means is that any shared traits that arise after they've split are due to convergent evolution. So I talked about convergent evolution. We're going to talk about that now in some more depth. Convergent evolution is when a similar trait evolves independently in two species. So in this case, the trait is the whole woodpecker lifestyle of smashing holes in the bark and getting the insects that are hiding underneath it. And that has evolved independently in the true woodpeckers and in this woodpecker finch that has to use tools in order to sort of fulfill that lifestyle. One interesting question is what kinds of traits lead to speciation? And there are a couple that are mentioned in the text, one in this chapter and one in a different chapter. One is the idea that sexual selection leads to rapid evolution in general and speciation. And so the question is, or the prediction is, do species with more competition for mates, so a mating system like lek polygyny, where a single male can mate with several females and other males don't mate with any, where there's more competition for mates in contrast to a monogamous mating system, does that lead to more rapid speciation? And there seems to be some evidence that it does. You have this example of these two sister species.
Less than 2%
Okay, we're gonna watch a video about birds of paradise for a second because they're awesome, and then we'll explain the example a little bit.
In the St. Island of New Guinea, there are 42 different species of birds of paradise, each more bizarre than the last. This terrace is so rich that nourishing food can be gathered very quickly. That leaves the male six-grooved bird of paradise with time to concentrate on other matters, like tidying up his display area. Everything must be spick and span. Very impressive. Something that unimportant. The superb bird of paradise calls to attract the female. And he has more luck. But what does he have to do to really impress her? She replies, consider her living. It's hard not to feel deflated when even the best isn't good enough.
Okay, so birds of paradise, fascinating, weird mating systems where they build these heavily ornamented nests sometimes, and they do these displays, and they display for all these females, and then all the females mate with that guy. And that's in contrast to, and if you were interested in learning more about birds of paradise, there's like this amazing website with lots of different displays and stuff. In contrast, there's a sister taxon, the Manicodia, that is monogamous and doesn't have a lek mating system. And there's just one species of Manicodia, but there's like 37 species of birds of paradise. So it's suggestive that species with stronger sexual selection with greater asymmetry of mating success among individuals are able to more likely to speciate more rapidly, which is kind of interesting. Okay, so I talked about convergent evolution, and so I want to, develop that definition a little bit more. Analogous traits are similar traits that have arisen in related or even unrelated groups of organisms independently. Convergent evolution, independently evolved traits that arise due to similar selection and look similar, even though they might have very different origins. So convergent evolution is, on the one hand, convergent evolution is like a problem for evolutionary biologists because you might think the two species are closely related because they share a trait, but if they share it due to convergent evolution, they may not be closely related, right? So it can confuse evolutionary relationships. On the other hand, Like if we discover that traits are convergent, now we know something. We know that there's something about the ecology of those species has strongly favored that trait. So we can look at the ecology of those two unrelated species and learn like why that trait evolved in the first time. So analogous traits that have the same function but different origins, are also sometimes called homoplastic. I'm just going to use analogous versus homologous. The opposite of analogy is homology. So I'm just going to, I forget if I write, this happens, like it'll be defined in 3 slides from here, but I can't remember that. In contrast, homology, homologous are shared due to common ancestry. Like, we've got 4 limbs, dogs have 4 limbs, salamanders have 4 limbs. Those are homologous traits because we all descend from a fish that had pectoral fins and pelvic fins that became modified into legs. Those are homologous traits, those 4 limbs in all the tetrapods. Convergent traits, in contrast, are traits that arose independently due to similar selection acting at 2 different times. So, example of convergent evolution are castes in many unrelated social species. So, honeybees have some very distinct, they're not super distinct, actually, there are some that are more, but you have queens that are females that mate with drones and don't do any foraging, don't do any guarding, don't do any feeding. They just eat and produce eggs. And those eggs are then taken up by workers that are mostly the daughters, maybe the sisters of the queen. And those workers then place the eggs in cells, they build the hive, they do all the foraging for nectar and pollen, they feed the young. And they're shaped a little differently. The workers have bigger eyes, smaller abdomen. The workers can only sting once and then they die when that happens because the stinger and much of their gut is torn out when they sting. Queen can sting many times because she needs that to fight with other queens when she's trying to take control of a nest. And the drones don't do any work. They have huge eyes because they have to be good at flying after queens to try to mate with them. So those are castes in honeybees, division of labor, especially reproduction. Only the queen lays fertile eggs. Yeah.
Just going back to homologous.
Yeah.
I saw a picture where it was showing the arm of like a human.
We're going to be doing that example in a couple slides. So wait till we do the example. Termites also have division of labor. They have separate castes. They have reproductives that, as you can guess, mate and reproduce. They have different kinds of workers. They have small workers that, collect wood and chew it up and harbor the symbiotic parabasalids and bacteria that digest the cellulose. They have soldiers with big old heads with large muscles, and sometimes they can squirt toxins, and they're shaped really differently. They're different than bees because there can be male and female workers, whereas in honeybees, all the workers are female. They're also not closely related at all. Bees are related to ants and wasps, and termites are related to cockroaches. And they don't share a common ancestor for like 100 million years earlier than either of them. So this whole thing about division of labor evolved independently in bees and termites, and you're like, yeah, I believe that. But you have the same system evolving convergently in a mammal. So there are naked mole rats that live in different parts of Africa. They've been studied well in Kenya that specialize, so they live underground. You don't see them that often because they do everything in burrows underground. And they try to find these large tubers that a whole colony can live on for a while. And then while they're doing that, they send out other scouts to dig new tunnels to find new large tuberous roots. And they have reproductives, just like an alpha queen and like maybe one or two other females that are reproducing and a few males. And then everyone else, like the termites, male or female, are workers that don't reproduce. What kind of maintains this division of labor in the mole rats is that they have sort of communal pee and poop chambers, and the kind of queen has like a hormonal signal when she's healthy that suppresses ovulation in the other females in the naked mole rat colony. So very similar social system evolving completely independently of bees or termites. Yeah.
So this similarity in behavior is considered convergent evolution?
Totally convergent, yes. The shared ancestor of mole rats and insects is going to be some kind of worm. Like these are protostomes versus deuterostomes, like 600 million years ago, 550 million years ago, their ancestors split. But now they have the same social system. Yeah.
So, do they communicate somewhere, or they just come out and like know what to do?
What? Naked mole rats?
Yeah.
They communicate a lot, and there's still a lot of instinct or genetic control of behavior. But like what's happening is it's the same thing in the bees. The ones that are reproductive have to constantly give usually chemical cues that they are in good health. And if they are not able to produce that signal, the workers get rid of them. Like in honeybees, if the queen is not laying eggs and not producing the right chemical smells, the workers will kill the queen, right? And it's the same in mole rats. So it's sort of a dynamic system that's being maintained by sort of a constant communication. Another example of convergent evolution is one of our most highly specialized organs. Not our phones. Our eyes. So eyes, complex. You got a lens, you got a cornea, you got iris, you got the retina. Eyes have evolved independently multiple times. How do I know? A range of lines of evidence. So we'll talk about this more when we talk about like mollusks, which is the classic example of a simple focusing eye. And when I say simple, I mean like insects have complex compound eyes. We have simple eyes with a lens that focuses. Our lens focuses by changing shape, and I think the mollusk, the squid and octopus eye focuses by like being moved. But what's really interesting is there's some really important developmental differences between mollusk, octopus eyes or squid eyes and human eyes that show that they evolved convergently. The differences include that in vertebrate eyes, the retinal cells project into the center of the eye. So the optic nerve, which starts off as the projections of all the light-sensing cells in the retina, forms in the eye and then actually has to kind of punch through a hole in the retina to get to the rest of the brain. And so that's why vertebrate eyes have a blind spot. Each eye has that spot where the optic nerve goes through the retina, and you can find it, hopefully you've done that in grade school or high school. You put a dot on a piece of paper, cover an eye, and you move your eye until you make the dot disappear. Why is it disappearing? Because it's now over the blind spot, and your algorithms of correcting and fudging over the blind spot correct for it, and now it makes the dot disappear. Octopus eyes project backwards. It's a better design. Often the best examples of pieces of evidence for evolution are the vestigial weird things that show that it could have been done better, but the reason why it's like that is because it had to work with what happened before. So mollusk eyes project backwards, they don't have a blind spot. The other thing that's different between mollusk eyes, there's a lot of differences. Mollusk eyes and vertebrate eyes is that they originate from different tissue layers. So I think the lens in mollusks it's ectoderm, and in vertebrates it's mesoderm. I am not sure, so don't quote me on that. But I know that there are different tissue layers of origin. Happily, I will have a chance to look it up and come back. It's amazing. After 2 hours, you forget this basic stuff. Anyway, when we talk about protostomes, deuterostomes, and chordates, we're going to come back to this example in a little bit more detail. But so developmentally, there are some different tissue layers that form the eye, and morphologically, they're quite different. But convergently, it's amazing. There's an iris, a lens, a retina. They're incredibly similar, showing that if you want to be a good eye, there are some really strong design constraints that are going to shape that convergent evolution. Okay, in contrast, homology is similarity due to shared ancestry. And this is where we get back to the textbook example. For example, the bat wing is a front leg that's been modified into a wing, and it is absolutely homologous to a bird wing or our arms. So the limb is homologous. However, the trait of flight is convergent. In other words, bats evolved flight completely independently from birds. We can see that in a bunch of ways. We can see that because the changes in form have been different. Bats maintain 5 fingers, and they have skin that's webbed between the fingers. Birds have fused, reduced down to kind of 3 fingers. 2 have been completely lost, and the remaining 3 have become fused, and it's the feathers that make the flying surface. But also, all the close relatives of bats are all the other mammals that don't fly. All the close relatives of birds are all the other reptiles that don't fly. And that's how we know that the trait of flight evolved convergently. So we have homology and convergence existing at the same time in this same trait. The limb is homologous. The trait of flight is convergent. So this is the level of homology. These bones are the same bones, but they've been modified, but the trait of flight has evolved independently. I'll throw in a third example, or the text will. Insects fly. The evolution of flight in insects completely convergent to flight in mammals. Insects and mammals share a common ancestor in a worm. But also, insects are not tetrapods. They don't have the same kind of walking limbs. Insect limbs arose completely convergently, analogously, separately from vertebrate limbs. And so the wings of insects are not homologous to the limbs of bats or birds, right? These actually probably arose as modified gills in the aquatic larva of an earlier flightless insect. And you'll notice the wings of insects aren't even modified insect legs, right? Because a flying insect has 6 legs and wings. A non-flying insect also has 6 legs. That's because wings don't come from insect legs. Anyway, so there you go. Insect legs and vertebrate insect limbs and wings and vertebrate legs or wings are completely analogous. They are not at all homologous. Okay, So, homology. One thing that's really interesting about homology, the last thing I'm going to end with, is that more recent studies in the last 20 years, 30 years, have shown that on a molecular level, we have a whole new set of questions about homology and analogy. And sometimes the molecular mechanisms underlying convergent traits are, in fact, homologous. So I made a big deal about how eyes evolved convergently, independently. They totally did. But some of the light-sensing pigments used in the eyes are coded by genes that are homologous. In other words, some of the molecules used in these eyes that evolved independently have a common ancestor, are homologous. And similarly, some of the genes that direct development in a lot of animals that are really different, like interestingly, legs evolve convergently, independently in insects and in vertebrates, but some of the genes that control segmentation and leg formation are shared. They're now using a somewhat different, they're using, they're leading to a different kind of segmentation and a very different kind of limb, but some of the same genes are being used to specify leg formation in insects and in vertebrates. So there's a good video on that, and I just uploaded it to G Suite. You should watch it there, not here. It's called Homeobox Genes. Okay, so that went a little longer, but I still have a bunch more slides. But don't leave, because what time is it? Kahoot time! Okay, let's see if this works. First, I want to close this. I'm going to close the lecture. Going to stop the recording. Let's see if that was working. Okay. Yeah, baby. I'm going to save this recording. It's working. Okay. So, hopefully that's going to work. And now, we're going to, this is a bit of an experiment. Oh, don't do that. I'm going to give this a try, but what this means is I'm only going to be able to record the audio if it crashes in the middle. I'm going to try audio. I can always record audio. Sometimes, like when it's too long or too big, but I'm going to try to record the whole thing a little bit later on. But it's recording the audio. Then I'm going to try recording a smaller video file. You can do it. Come on, we can do it. Okay. Okay, so I successfully recorded audio. It's saying it's going to take 23 minutes, which means it's eventually going to say it's going to take 100 hours, and it's never actually going to work. But while that is failing and making me sad, let's try doing a Kahoot. So I think I just click play. And so use your ID for a nickname. and we're gonna do classic. Whoa. And we need the pin. Let's make this quieter. 35097. This is better than the version we played at my wedding. I got 6 players. Is that it? Now it is. All right. I got 20. I feel like we have about 110 people here, so that's what we should try. Okay, is everyone in? Okay. Sorry, what? Is it not working?
Well, so I think what you added over there, the
Yeah.
pattern,
Yeah.
it's a little bit of a barrier because like some people had like trouble like going on time without it when Dr. Alvarez did it.
Okay. So I can do a setting where I don't ask
Yeah, I
for the
think
pattern?
you could like just not have the pattern, just have the pin, and
Okay.
you'll have no problem.
Great. I'll try that a few times.
Yeah,
for next time. Okay, are we all in? Okay, take your time. I'm sorry. I'll figure out how to do it without the pattern. How did we lose a player? Okay, are we ready? We're gonna try it. Here we go. Okay, islands separated by oceans can lead to allopatric speciation, phylogeny, anagenesis, sympatric speciation. Woo! Okay, that's good. So, islands allopatric, different areas, islands separated by ocean can lead to speciation, allopatric speciation, Darwin's finches, Hawaiian Drosophila. Okay, let's do it again. Oh, I have no idea what that means. Okay, the forelimb of a wolf, bird's wing and bat's wings are... Hopefully this one will be really straightforward. We just did this. Okay, so they're the front legs of a wolf, the bird's wing and bat wings are homologous structures. They are shared due to common ancestry. They're all derived from the pectoral fins of the amphibian fish ancestor of all tetrapods. Okay, cool. Sympatric is nonsensical.
I crashed.
You crashed? Ugh. Okay. Well, I'll still have a record for that first answer. So, in terms of participation, you're fine. In terms of playing the next game, it's a problem. Okay, the evolution of flight in birds and bats is, these are easy because they're reinforcing what we just did. So the structure is homologous, but the trait of flight great. So, but know all the terms, right? Cladogenesis is when a taxon splits into two separate clades, two species, or one family splits into two families. Divergent, no. Homologous and convergent are kind of the two opposites, and it's convergent. And we know that because the traits differ structurally and developmentally, like how the fingers are modified versus fused and reduced, and like all the close relatives of birds. Other reptiles don't fly. All the close relatives, mammal relatives of bats, mice, foxes, beavers, they don't fly. So we know the trait arose independently. Yeah?
If let's say two species converge like so much, can they become one species?
So the traits can become convergent, but if they're really two species, they're not going to be able, and they're very unrelated to each other, they're not going to be able to mate with each other, right? So traits can converge. Two species, what's called, if they were like related to each other already, and they were different, and then they start hybridizing, and it's called fusing. It's not called convergence. Okay, next. I have no idea what those numbers mean. Species that look alike but avoid mating due to differences in timing or signals used are... So this is not harder, it's just an hour and a half old. Okay, so this is good, right? This is good. We look at this and then we learn and then we get it right on an exam. This wasn't so much harder, it's an hour and a half old. Confused with more recent things. Species that look alike, so you'd think they're the same species, morphological species, they're the same, but they don't mate with each other because they have different calls or different timing. Those are cryptic species, right? They're not necessarily sympatric. Just because they look alike doesn't mean they live in the same area. They're cryptic species. Okay, awesome. That's good. That's why we did this. If everyone got them all right, there would have been no reason to do this. Okay. Last one. Sometimes populations or species found in the same area are more different than those two populations when found in allopatry. Cool. Yeah. So reinforcement is the process. Selection against hybrids is the process that will make species more different. Cool. Okay, I think that's... I have no idea who these people are, and that's fine. But that's great. And like I said, in terms of points in the course, it's just participation. But in terms of knowing the material, it's good feedback. So that was awesome. That was so much more fun than the Blackboard quizzes. So I'm glad that Professor Alvarado introduced the system. Let's take just a couple minutes, and then I have a phylogenies lecture. So take a couple minutes, stand up. Yeah. My respect. The feel of your peers. Yeah. Okay. So here's the thing. You send me an email. Obviously include your user ID. And I have to go into this and I'm going to download it as an Excel file the results. And I'll just see if it shows up.
And also, I also do not have access to Google Docs, which I've tried to get access to, but I can't.
Okay, so that, yeah, you should send me an email and I can get you access. What I'm going to do is I'm going to try restarting my computer so that the lecture recording works. But, so I'm not going to do it right now, but you should write to me. And yeah.
I have the same question. So,
Yeah.
like, I did it, but nothing was like clicking or moving forward. And then I did it from my phone, and then it didn't work on my phone either. So do you want me to send it to you?
Yeah, send me an email. Yes, sure.
With my Apple ID.
Not Apple ID.
No, Android.
Your email.
Yeah.
Yeah.
Quick question from the lecture.
Yeah.
From the reinforcement, the isolated south and the southern population,
Right.
they got distinct.
Right. They became more different because the isolated southern is that little island surrounded by northern, and they're able to hybridize with the northern.
So to avoid that,
To avoid that, they become more different.
are they
Yeah.
considered different species? Or they can still
No, it's like subpopulations or subspecies. They're like on the way to becoming. And the truth is, like, people argue about whether there are so many examples, so many people are studying, and people always argue about are they different species or not, and they don't.
they can
So,
still have the fixation.
yeah,
Okay, got it. Thank you.
yeah. Okay. What's your name? Okay. So we got a bunch, and I thought we wrote back to them, but I'm not sure. So what's your constraint? If it's multiple exams the same day, we have like about 10 students. What was your?
So I registered for
Oh, I remember. Yeah, I totally remember that. So that's right. You have a travel thing,
Yes,
right?
and I can bring it over from my job.
Yeah.
I can even take the test before, so it's not like I'm
Yeah.
tired
What time can
anymore.
you take it on? It's next Thursday. You said you
Can I
can
take it on the
Wednesday.
16th? I can take it on Wednesday. And I know we have a Monday schedule that day, but I can
Right.
take it, I leave Wednesday at 10 p.m. so it's good at 8. So I can take it.
So when is your flight?
I have to be at the airport at 8 p.m. So let's say I guess the latest I can
8
take my
p.m.
exam.
on Wednesday or 8
Wednesday.
p.m. on Thursday? Okay. Yeah. Send me. Or show me your travel set.
Okay. I have to have my job send it to you to have any questions.
Right. So we can do it Wednesday afternoon.
Okay.
I'm not sure if it'll be in my office or Dr. Robert's office. I'll probably set you up in another.
Okay, so do you want me to email you again about this? Or
Yeah.
just go there? and
Yeah, you should, because emails get buried. I remember your email. I feel like you can send you. You can move it later. Yeah.
I'm really annoying. I know that we emailed you about this, but it actually turns out I have 4 exams that day. And she as well, we have all the same classes.
What are the 4 classes?
3 of them are sciences, organic chem 1, bio 106, psychology, and Russian.
Okay. So, what did your professors and all those other courses say?
I mean, I ideally wanted to split up Orgo and Bio, considering those 2 are the hardest, and I spoke to the Orgo professor, and he said he wouldn't be able to do it. So you're my last hope.
Yeah, I mean, I think I talked to Professor Alvarado, and he didn't want to move it, and I didn't either. And it's kind of frustrating. Like if it's for the final exam, if we have 3 at the same time, we move it. If it's 2 the same day, we don't. But for the midterms, it's really hard. It's hard to find a place to take it, and it's also just frustrating once we have a bunch of students doing that, because questions come out and we usually don't have 2 exams that are, or a 3rd exam that's as different as the 2
It's
that
just
we're
that,
doing.
I don't think that it's I know it's obviously not your fault, but I don't think it's fair that I won't be able to do as well had it just been that exam or just 2 exams. 4 exams is a little excessive.
Right. Yeah, I mean,
And anyway. I could take it the following like Tuesday, the next lesson. I know in Bio 105 they would be like redoing the exams for like 2 weeks because like people were like out sick and stuff along those lines.
So yeah, I mean, I'm sure that I'm going to be having some makeups for students that are sick. We get like 1 to 3 of those every exam. It's different because in theory you're able to like plan for this and study. I mean, I know it's frustrating. Yeah. How many of you are there?
I know there's a lot of people taking both Orgo and Bio 106 together. I think probably at least like people that I know, maybe like 10 to 12 people, and that's the people that I know. But then like for the 4 exams, it's me and her.
Yeah. That's everyone here? No.
Orgo Bio.
Sorry, what?
Orgo and Bio.
Orgo and Bio. Yeah. Yeah, I think we're not gonna do it. I think we're not having a second exam. So we still gotta take it. I'm so sorry. Yeah, I mean, come up again at the end of lecture, but it's probably not going to happen. I know it's going to happen because I've been talking to Dr. Alvarado, and he'll be like, no, we're not doing 2 exams. Yeah.
I mean, is there a difference, like, with, like, why, could we, like, take it with the people that are, like, gonna be out sick? Because there's really not much of, like,
I think the idea is that when you can plan ahead, that you try to not, well, practically, we just try to keep the number of exams other than that first period to the absolute minimum because once we give an exam, students talk, which is fine, and then like it's no longer a meaningful exam. Like your exam is going to be different because you're studying for 2 exams at once, but if you took it the next week, it's going to be different because you get the feedback of students that have already taken the exam. And it's true, the students that are ill also have that, but this one's preventable and that one isn't. So that's why we do it. One thing that I often do when I'm teaching the class, and I'm happy to do this time, is that if students do 10% or more better on the final than they did on the first midterm, I'll count the final for 30% in the first midterm for 10. So I'll count the final more. So if this undue, normally that's just to keep people from giving up hope in the course. It's something that I would do for the whole class. But if your first midterm is screwed because it's impossible to study well for both of them, then you'll do better in the final, and you won't be penalized in the same way.
But I mean, if you want an A in the class, all those little points count, even if it's only going to count for 10%. Well, is there any way you guys can take it?
That's true. But, we usually, despite... That is true, but it's only a small fraction of the class that's...
It's not just 2 exams, it's 4 exams. Like, it's 4 exams. Like,
yeah.
I get that it's like you can plan ahead, but at the end of the day, like, the most effective, like, for a certain, like, it needs to be fresh, Like, I could do it 2 weeks ahead. It's like, it's not going to be as, like, effective had I done it for just, like, the one class, like, a couple days before.
Yeah.
Anyway, you can make it Tuesday.
Did you take it, like, the day before? You mentioned to the other girl who was here, like, she could take it.
Yeah, I'm trying to prevent that from happening. Like, that other student was able, everyone's different, But that one student was able to show that they'd been dropped from the course because of the registrar. They had a problem with the bursar, and then they made this travel arrangement earlier, and then the bursar, they paid their bill, and the bursar put them in the course. And it's sort of unique. I sort of regret saying it's okay for that one student, but doing that for 10 students is different for 10 students. Anyway. Right to Dr. Alvarado and me again. We're gonna keep going, but yeah.
I spoke with Dr. Alvarado. He said that he didn't really have a problem with it. That he was just gonna kind of like check with you, and if you were okay with it, then
I will be talking to Dr. Alvarado. That's not what he told me. So yeah, we'll go back and forth.
Also, would you happen to know if there's any kind of like maybe like departmental rule against taking a certain amount of exams in one day?
So actually, I'm not sure. At UC Davis, where I was teaching a long time ago, there was the rule that like if there were 2 finals, you didn't reschedule. If there was 3 finals, you do reschedule. That's what I do. I'm not actually sure if that's a Queens College rule or if that's just like what I do because that happened before. But that was for final exams. For midterms, I usually never reschedule. I once rescheduled. It was a different conflict. It was like a Jewish holiday thing, and I put it to a vote in the class, and it was like 85 to 15 in favor of rescheduling. Because that's the thing. Like 10 of you contacted us, but in the past, when I've then put it to a vote in the class, it's like 55-45 or 60-40. In which case, I usually don't change it. And in this case, it came to us a little bit late, and we teaching differently, so I didn't have time to bring it up for a vote. And now we're the week before, so I'm not rescheduling. The question is can I do a different time for you? Still saying no. Okay.
Different question.
Come up at the end of class.
I was a minute late. Did you tell us the format of the test?
What?
Voice?
Oh yeah, I'll announce that for the whole class. Yeah.
Thank you.
Sure. Okay. (106). Try again. Take 2. And okay. So a student asked just the format of the exam. If I didn't mention it before, 60 multiple choice questions. I'm not 1,000% sure because I didn't write this one, but I'm 99% sure. No calculator.
No calculator.
Yeah. Both. Bring in pencils. Bring in multiple pencils. Multiple choice. Scantron. We provide the Scantron, but just expect it. Okay. So, second half is phylogenies, how to make them, and what they're useful for. And so the first part, actually, you probably went over in lab, is just this idea of how much information is nested in sort of the systematic classification levels. And that is because all of biology is nested, right? Because of shared ancestry, all of life arose from, as best we can interpret, a single ancestor. Shared DNA code, stuff like that. And so a great deal of information is encoded into the taxonomic hierarchy. So you did something like this in labs. I'm going to be really quick. But if I say that, I studied Passer domesticus, that might not tell you that much. But if I told you that I studied an organism in kingdom Animalia. Well, now you know a bunch of things. You know it's multicellular. You know that it's a heterotroph. You know that it has no cell wall. You know that we'll learn a little bit more. You know that it's got development guided by Hox genes. It uses collagen. There's all sorts of stuff that you know just because it's an animal. If I told you it's in phylum Chordata, now, bam, you know that it's got a dorsal hollow nerve cord, pharyngeal gill slits, a notochord, and a post-anal tail, at least early in development. So that tells you something. If I tell you it's in class Reptilia, subclass Aves, now actually you'll know it's got feathers, it lays eggs, it's got 4 limbs, it's got an amniotic egg, it's a bird. If I told you it's in the order Passeriformes, that actually, now I've eliminated half the birds. It's in the single order with the most species. It learns its song in a similar way to humans learn speech, which is really cool. We'll talk about that in the animal behavior lecture. It's got a syrinx. It's got some specialized grasping muscles in the feet. Those are all Passeriformes traits. If I told you it's in the family Passeridae, so sorry, this is the songbirds. Passeridae, well, now you'll know that it's an old world seed eater. That's not an insult for your family. You're an old world seed eater. Anyway, it's got, a bill that's sort of conical for crushing seeds, opening seeds. If I told you it's in genus Passer, actually I no longer know what that means. It's, I think there are 13 species in it, but I don't know what different species are in the genus anymore. But species Domesticus like domestic, domicile, home, it's a house sparrow. Cool. Introduced in the US, now fairly common, adorable. And there you go. So there's a lot of information that's nested in each of those levels. Similarly, we did this exercise one more time. Apis mellifera, Animalia, same thing, multicellular heterotroph. Arthropoda, jointed appendages, segmented bodies. Hexapods, it's an insect, has 6 legs. Hymenoptera, it's in the bees, ants, and wasps. It is haplodiploid, so it has that weird genetic control. Males are haploid, females are diploid. It's got a thin waist. It's got a bunch of features. Apidoidae, it's a kind of bee. Apis mellifera, it's the European honeybee. So there's like a lot of information that's nested in each of those taxonomic levels. And that's all I'm going to talk about that. You talked about it some in lab. Taxonomists try to understand the correct patterns of ancestry and descendants. And they do that by defining what are called monophyletic groups. And we're going to define that a couple times, so I'm not going to write it down here. Monophyletic group is one that contains all the descendants of a single common ancestor and that ancestor. So there are actually a bunch of possible monophyletic groups in this phylogeny. The crocodiles and the birds, that's a monophyletic group. So are all the reptiles, as long as they include birds. So are all the tetrapods here. These are monophyletic groups. What's not a monophyletic group? If you just group them, the warm-blooded animals. So if you lump the mammals and the birds together because they're both endotherms, and so they both have a 4-chambered heart, you'd actually not have a monophyletic group. You'd have a group of species that are not that closely related, where members of that group share common ancestors with other organisms like crocodiles, snakes, and lizards more recently than they share with each other. So monophyletic group, all the descendants of a shared common ancestor. We'll get to go over that a couple more times. A lot of definitions in this lecture. And for understanding these phylogenetic trees, these branching diagrams showing common ancestry. So systematics is the name of the whole study of the shared ancestry of taxa, the relationships among organisms, species, families, phyla, etc. A phylogeny, or an evolutionary tree, is a diagram that represents the patterns of shared common ancestry. So there's different ways of drawing them, but always the branching reflects cladogenesis, splitting along time, and so the further down or the further to the left in a phylogenetic tree, the further back in time, the older. A taxon is any named group of organisms. So a taxon can be a species, but a taxon can also be a family, a genus. A taxon, really you can do gene trees as well. A taxon is any named group of organisms. The tips are the descendant taxa. The nodes are the branching points. And a clade is a group. A clade is a monophyletic group. It's a group that includes a common ancestor and all the descendants. So a clade is a monophyletic group. Sister groups or sister taxa are two taxa that share a common ancestor, most recently with each other. So basically, it's a monophyletic group of two taxa. Those are sister groups. So you can see all of that here. Just, to point out, and these figures like this are in the text with like different species. Those are tips. These are nodes. These are taxa. Here's a clade. And in this phylogenetic tree, crocodiles and birds are sister groups. I want you to feel really good about these definitions of monophyletic groups, paraphyletic groups, and polyphyletic groups. And this figure is in the text, so totally go to the text and puzzle over it, because I could possibly, and by that I mean I will, show you a figure like this on the exam and have something where it's we could go A, B, C, and ask which one is the monophyletic group, which one is the paraphyletic group, which one is the polyphyletic group. So you really want to feel good about these groups. Monophyletic groups I've already defined. All descendants of shared common ancestor. So which is the monophyletic group? I wish I could do it in delightful Kahoot form, but yeah, it's C. Here's a common ancestor, all the descendants. That's a monophyletic group. What about A? Excellent, because we're getting to all of them. Don't worry, we're not skipping them. A has a bunch of descendants of a shared common ancestor, but not all of them. It's missing this guy. This guy's not in the group because this shaded box is the group. That is called a paraphyletic group. And that has some, but not all, the descendants of a shared common ancestor. And B is just crap. Like B, this taxon shares a common ancestor more recently with everyone here in C than it does with anyone in these taxa. These two taxa share a common ancestor here with all of these guys in A and then little orphan Bluey over here. They're all more closely related. So a polyphyletic group is one where members of the group share common ancestors with taxa outside the group more recently than they do with each other. So another way of thinking the cartoon version of a polyphyletic group is a bad group or a group of unrelated taxa. So polyphyletic, monophyletic are kind of the easiest ones to recognize. Paraphyletic, somewhat confusing because they all the members share a common ancestor, but not all the descendants of that ancestor. A paraphyletic group is like an incomplete group. Monophyletic is the complete group of descendants of a single ancestor. Polyphyletic is an unrelated mishmash. Okay, so we're going to talk a little bit about what kinds of traits are used to define groups, and then we'll get back to that bird example because it's very informative. Are birds reptiles? Are birds not reptiles? That helps sharpen our understanding of paraphyletic groups. Okay, so how do you figure out these phylogenies? You look at a whole lot of traits and how they're shared, and you choose the phylogeny that requires the least number of changes in traits to get from an ancestor to all the descendants. And that's called parsimony. Parsimony, like the dictionary definition of parsimony is like being miserly, not wanting to spend a lot. And in this case, it's instead of not wanting to spend money, it's if you're parsimonious, you're not saving money, you're saving evolutionary changes. Because the idea is that those changes are rare, and so the most likely phylogeny, a relationship of, evolutionary tree, is the one that requires the fewest number of changes. So we use parsimony to compare first the taxa that share the most derived characters. So derived means, different, it's written. Fantastic. Derived means traits that are different from the ancestor. Those are the most informative. It's not just how many traits do two taxa share, because they could share a gazillion traits, but if they share them all from a common ancestor, that doesn't tell you that much. But if they share a few traits that are different from the ancestor compared to other taxa that don't have those traits, now we know that those two taxa that share those derived, those different traits, are more closely related to each other. Even though both of those taxa may share a gazillion traits with the third one that they all share from the common ancestor. It's the derived ones. It's the new traits that are shared that show evolutionary relatedness. But how do we know what the ancestor has? And we use that by figuring out an outgroup. So an outgroup is a taxon known to be less related, known to share an ancestor more distantly in the past. than any taxon in the in-group. And the idea is we use that out-group as our best guess or approximation as for what the ancestral traits are. And so the idea is that if any of these taxa in the in-group share traits with the out-group, the assumption is that they share them because they all came from some common ancestor that had that trait. That's the ancestral trait. But if any of these taxa are different from the out-group, the interpretation is those traits are ones that are derived, are different from the ancestor. So in this sort of vertebrate example, lampreys, they don't have jaws. They're sort of jawless fishes. We'll learn about them later. They're sort of ectoparasitic fish. And all of these guys have jaws. So that's a trait that arose after the outgroup split off. But some of these groups share traits only with each other and not with perch or salamander or lizard or crocodile. And so those taxa are grouped together. So for example, mouse and chimpanzee share all the mammal traits like fur and mammary glands. That's actually really all the mammal traits that there are. Like live birth happened after mammals split off because like platypuses still lay eggs. Anyway, and so, but they share those traits with each other that are different than lamprey. None of the others have them. These guys are assumed to be most closely related to each other. So you assemble a cladogram by first counting up all of the derived traits, and then seeing which taxa share the most derived traits. And we sort of group those most distantly from the common ancestor. So here we have a bunch of vertebrae, and in this case, the lancelet is our outgroup, and the lancelet doesn't even have doesn't have a vertebral column, doesn't have a backbone. And then we're coding the traits as, if it's the lancelet version, it gets a 0. So we put zeros for all of these. The lancelet doesn't have any of them. And then if it has these traits, and that's going to be different than the lancelet, that's the derived state, it gets a 1. And so then we have all these traits: vertebral column, jaws, 4 walking legs, amniotic egg, hair. And what you can see, and then you score the different taxa for the traits that are shared or derived from the ancestor. And when you do that, you have your little table, and you can count up the number of shared derived traits. And so, for example, obviously it doesn't, all of these, share the same number with themselves. That's not the point. Mammals and reptiles share amniotic egg, 4 walking legs, jaws, and vertebral column. So that's 4. Mammals share only 3 of them because amphibians don't have an amniotic egg. Again, remember, mammals, you think amniotic egg, painful live birth. But the basal mammals, the platypus and the spiny echidna, the monotremes, still lay an amniotic egg. So we know that that is actually a mammal trait. The loss of that egg happened after mammals split off from reptiles and amphibians. Reptiles and amphibians also share 3 derived traits. Turtles and salamanders share 4 walking limbs, jaws, and vertebral column. Mammals and fish only share, I think, 2 because we don't have the 4 walking legs, and we don't have, read my mind, tuna, yes, jaws and a vertebral column. Reptiles and fish, also 2. Amphibians and fish also 2. Mammals and lamprey only share 1, that vertebral column. Reptiles and lamprey also share 1. Amphibians and lamprey, again, only has this 1 as well. So reptiles and mammals, they share both the vertebral column, fish have jaws, walking legs, and hair, amniotic egg, and then mammals are derived because they're the only ones with hair or mammary glands. But if you combine the 2 that share the most derived traits as the ones that share common ancestor most recently, and then you add on the other taxa sort of more distantly related. And the other take home for all of these phylogenetic trees is to understand that like time is one dimension of the graph, and we're always going from older to more recent. Okay, let's get back to the birds question, because it shows that that's a ridiculously easy example that's not always intuitive. So one question is, do you want to use highly adaptive traits or traits that were shaped strongly recently by natural selection? Actually, no. What you really want are neutral traits that are not affected by selection and evolve at a uniform rate. Why? Because those are going to accumulate changes consistently over time, right? When you learned about the neutral theory of evolution, what was that? Neutral theory of evolution is that most of the changes in gene sequence or base pair sequence are not shaped by natural selection. That's because most of the genome doesn't even code for, traits. A lot of it is like the remnants of retrotransposons and viruses and selfish stuff that have been like turned off, but they're still dragged along, and they evolve neutrally, like random genetic mutation and genetic drift. And then there are other mutations that are synonymous, that don't change the amino acid being coded for. And all of those can change sort of at a uniform rate over time. The traits that are non-synonymous, most of them are gotten rid of by natural selection because they usually lead to loss of function. Right? So most of the genetic change over time is neutral. Those can be used really nicely to build these phylogenetic trees. You get into problems when you start using traits that might have changed fairly recently, and by fairly recently I mean like 150 million years ago, by natural selection. Why? Well, selection can lead to a whole bunch of traits changing convergently because of sort of shared ecological requirements. You might not think about that, these traits as convergent, but birds and mammals both are endotherms. They maintain a very high metabolic rate, and it's an interesting question why. Birds need that high metabolic rate to fly. Mammals need a high metabolic rate to like colonize and be active in cold weather. And because of that, they have some traits that are shared. An insulating layer. I mean, it's different, but it's insulating. Fur, feathers, a 4-chambered heart, rapid growth, wide range of active temperatures, occurrence in the tops of mountains in northern and far northern and southern areas. For this reason, people used to think that birds and mammals were more closely related. They're kind of similar. Like, they're really active, lizards and snakes, they kind of sit there, and for a long time, birds and mammals were thought to be more closely related because they share a lot of those traits. But in fact, birds are good reptiles. They're highly modified for flight, but they are reptiles. If you look at gene sequence, if you look at the fossil record, birds are clearly more closely related in terms of when they split off from the phylogenetic tree to some reptiles like crocodiles than even crocodiles are related to turtles and snakes, some other kinds of reptiles. So this is a phylogenetic tree that has been constructed based on a wide range of traits. DNA sequence, protein sequence, much more subtle morphology than like insulating layer that's totally different. Things like, the structure of hip bones and internal anatomy. Birds are actually not just more related to reptiles than they are to mammals. They're more related to this small subgroup of reptiles that includes the crocodiles. What kind of group would reptiles be if we only included the scaly, cold-blooded things, lizards, turtles, and crocodiles? Paraphyletic, exactly. They're all derived from a common ancestor over here, but they're missing some of the descendants, the birds. And that's why, many evolutionary biologists, like me, call birds reptiles. Aves used to be a class, like reptiles and mammals and amphibians. Now it's a subclass. It's a subclass of reptilian. So, there you go. Okay, why do we build all these phylogenies? Because phylogenies, if we have a good phylogeny that's based on good traits, a whole lot of them DNA, proteins, not just morphology, now we can ask evolutionary questions. And these are called comparative methods. So we can make comparisons among taxonomic groups. We can ask questions about how evolution happened. So we can use a phylogeny to ask, did a trait arise just once, or did a trait arise convergently many times? We can also ask, do some traits lead to other traits evolving? So I'll give examples of each of these. I talked about a comparative method. We had a comparative method study before. It was does sexual selection lead to speciation? And the answer is, from the birds of paradise example, maybe yes. Because the birds of paradise have a whole bunch of species, like 37 species. Their sister taxon, it's killing me, it's in the Latin, it's gone, has just one species. The monogamous one has just one. The one with polygynous lek species mating system, where males display to many females and some males get to mate with lots of females and some not. Sexual selection much more important and much more elaborate displays. That has led to speciation. This is a better example because it's insects, and all insect examples are better. But also because insects are much more diverse, so you can actually do science. You have much larger sample size. That's what I mean by better. Not like they are better too, but because you have a larger sample size. So this is asking whether eating plants leads to diversification, more species, versus eating animals. And there have been a number of these comparative studies, and the answer is generally yes. So this is also a sister group, sister taxon comparison within the hemipterans, the true bugs. We're going to learn about them when we do insects. They have piercing, sucking mouth parts. They're awesome. So this sister pair, the Geopesids, are predaceous. They eat other insects. There's only a single species. The Tingids are also hemipterans. They eat plants. There are 1,800 species. Similarly, the isometopids are a group of true bugs that are predaceous, eat other insects, 60 species. The mirids, plant eaters, they have 10,000 species. Why do you think that might be?
Just more plants.
Yeah? More plants to eat? What do the plants do when they're getting eaten? They fight back. Yeah, so it is sort of related. There's a whole bunch of plants, and one of the things plants do as they're getting eaten is they evolve defenses. We're going to talk about some of those defenses. Maybe you got to talk about some of them already, but they mimic insect hormones. They have molecules that break down enzymes that break down insect digestive enzymes to give the insect stomach aches. They have all sorts of different toxins. They have like lignins that are just super hard to chew on. Plants evolve defenses. And what happens is for insects to be able to overcome those defenses, they have to specialize. So plant eaters, over and over again, evolve specialized strategies to overcome the plant defenses. That leads to speciation. It also leads to the plants evolving. They sort of co-evolve that way. Meat's meat. Meat's easy. Actually, you know that as a person. If you become vegetarian, you learn, I have to work a little harder cooking because, frankly, I have to overcome these plant defenses of, boringness and chewiness. Whereas meat is like, you heat it up and it, has its umami or whatever. It's just easy to cook. It's like any brain dead idiot can make a hamburger. But there you go. Plants are harder. And that is reflected in speciation specialization. That doesn't mean that it's not a great thing to become vegetarian. It's just, I find it's a little more work. Okay, so that's one example of comparative methods. We can now see, in some ways, speciation being more common in herbivorous versus predaceous insects. Comparative methods also allow us to ask questions about the origins of traits. And so this is an example, it's in the text. Reproductive success in male swordtail fish is associated with having a really long ornamental tail. And it's really weird because the tail makes them swim badly, but the females like it, the males grow it, it's kind of weird, they get eaten. And one thing is one of the fundamental questions of sexual selection is why do these weird traits evolve? And so one possible explanation is that they evolve because there's a pre-existing preference among females for some kinds of traits. And so this question has been asked using phylogenetic methods. So phylogenetic analysis, DNA, morphology, identified platyfish as the closest relative of swordfish. They're all actually somewhat related to guppies. Platyfish, the sister taxa to swordfish, but they don't have tails. So I think it was Basolo. Someone did the logical experiment. They made artificial swords. They made little swords and glued them to attach them to platyfish males, and then had tanks with dividers that the females couldn't actually, were stuck in the middle, and were given a choice between a platyfish without a tail and a platyfish with a tail, and you have a platyfish female in the middle, and she'd go over and likes to swim next to the male with the long tail. It's really interesting. So female platyfish preferred males with the fake swords, supporting this idea that a pre-existing preference on the part of the females biased selection towards this ornamental tail. So, for example, this is a male swordfish, and this is a platyfish without the ornament. And so, very simple phylogeny, but you can see that in this case, the preference is shown both in the platyfish and in the swordfish. The preference evolved before the sword. Actually, though, as often the case in these examples, there's some greater complexity. because swords are found in a bunch of species, some that are really closely related. So that means that all of these guys come from an ancestor, most likely by parsimony, that had a sword. But like none of these guys have a sword. But these guys have a sword. So there's more evolutionary questions actually than are answered. It's likely that the sword can sometimes get lost as well as arising, because the most likely parsimonious scenario here is that it was gained here and then like lost here and maybe lost there as well. That's the least number of changes on this phylogenetic tree. So now we still have this question, why might this trait of long sword tails get lost? Which is another interesting evolutionary question, but we only know that's a question because we have this phylogeny. And now we can say, oh, it looks like this trait arose here and was lost. Yeah?
So could this also be explained by convergent evolution?
Yeah, totally. It's also consistent with none of these guys having tails and the swords evolving independently in this group and in this group. But then that means what's similar about this group and this group that leads to both of them convergently evolving tails. So both of those are possibilities, and both of those kind of need to be explored further. You only get those questions when you have the phylogeny. Yeah?
But if the swords only came because of like the sexual selection, would it have to be something in the selection process that might have changed?
Right, well, it might mean, I can throw out a bunch of stories, none of which I've tested and stuff. It's possible that sexual selection is favored more in this group and that group. Why might that be? Maybe it's because the water is cloudier. That's been shown to be important in sexual signaling in fish. In those cichlids, you have brighter colored signals in water that is more often turbid, more often stirred up by dirt. You need a stronger signal for the females to be able to recognize it. Predation also often has a thing. It's possible that there's more predation here. So males with long tails are much more likely to get eaten, so the cost of having that signal is greater. But yeah, the phylogeny shapes the questions, and then you can go out and try to answer those questions. But if you didn't have the phylogeny, all you know is, oh, there's a lot of species that have tails and a lot of species that don't have tails. Okay. An example in the text, in the 11th edition, is the evolution of the long tail trait in Luna moths. So it turns out that these long tails might actually be not a sexual signal in this species, but an anti-predator escape response. So the idea is that if you have the long tail, your flight pattern is a lot less predictable and weirder, and it's harder for bats to catch you. And that is seen in flights in dark rooms in laboratories, where you set these moths flying and you cut off the tails of some, and the capture rate by bats is greater if you cut off the tails than if you don't. Really cool experiment. What the phylogeny has shown is that this trait of super long tails in Luna moths has actually arisen multiple times independently. In this species and in Eudaimonia argus and in Castionacera hercules, that have a whole bunch of relatives that don't have a long tail. So this trait has arisen multiple times, or maybe it's been lost multiple times. More complex phylogenies have shown this in greater detail. You see it in fairly somewhat related, but also sort of unrelated, showing that we need to know a lot more about this trait. Is the bat predation really different in species that have it or not? Did this trait evolve really independently, or was it lost? So you need phylogenies to show that. Okay. All I'm going to say is that manipulative experiment has been repeated on a whole bunch of different species and shows within and among species, the ones that have longer tails are better able to escape bats, which is really cool. But now, like, that opens this whole new question. What among these 30 species, separate from tails, makes the bat selection stronger or weaker? Okay, so the take-homes from phylogenies. Systematists want to find monophyletic groups because monophyletic groups show the correct pattern of shared ancestry and descent. We build monophyletic groups by pairing together the taxa that share the most derived traits. And remember, we use an outgroup to understand which traits are derived, new, versus ancestral. Once we have this phylogeny, we can ask evolutionary questions like, did a trait evolve once or many times? Is it easily lost as well as easily gained? And then maybe what about the ecology affects its trait, like eating plants versus eating animals? I wanted to finish with a really short sort of coda on what races are, partly because these phylogenetic trees can also be shown to look at genetic similarity among human populations and other primate populations, and also because we've been talking a whole lot about subspecies, and if you're an evolutionary biologist, you'll read about some biologists will talk about races of butterflies or birds. Is that the same thing that we mean when you read about races in humans? And the cartoon take home is they're not. Evolutionary biology definition of race is very different than sort of socio whatever definition of race. Evolutionary biologist's definition of race is kind of what we would think of, what I've been using to say synonymous with subspecies. Ernst Mayr defines a race or a subspecies geographically distinct populations with different characters. And evolutionary biologists, when you're talking about plants and animals, you're usually thinking of races as kind of on the way towards speciation. Is that, and the important character is that races are populations or subspecies that are differentiating. They're probably on the way to becoming different species because of that geographic isolation. Easier to recognize if the species is sedentary, if individuals don't migrate. It's easier to arise too. Migration opposes, differentiation. We talk about the example of Rhagoletis pominella, the apple maggot, hawthorn maggot fly. Those are kind of on the way to becoming separate species. So how does this concept apply or not apply to humans? There is genetic variation in humans. There's a lot of variation. We look around the room, we see a lot of variation when, diseases pass through populations. Some individuals are more affected than others. The level is humans vary on average, and it is and was a valid empirical question. How much genetic variation is there among humans? We vary on average by 1 nucleotide per 1,000 nucleotides. Some variation among humans is likely adaptive. And I will point out that I'm going to mention these 3 examples. I can't think of many more. There haven't actually been a lot of adaptive traits identified among human populations that differ. One example is the ability to produce vitamin D. How much sunlight do you need to produce vitamin D? And that is associated with melanin. And it varies among populations that have consistently differed in the sort of average amount of sunlight and day length. And it's a trait that's evolutionarily plastic. In other words, it has evolved and changed multiple times over the course of human evolution. Malaria resistance is another example. We'll talk about that again, or maybe you talked about it already with Professor Alvarado, because it's a classic example of heterozygote advantage, right? The optimal trait is to have resistance to malaria without debilitating sickle cell anemia, but that only happens in the heterozygote form, so you end up having both traits maintained for a long period in the population. Another is lactose digestion, which interests the ability to digest milk sugars in adulthood, associated with cultures that cultivate cattle. Another trait that's emerged convergently multiple times. Really cool. It's been seen to evolve. It's evolved within the last 5,000 years, multiple times, which is really cool, through different genetic and epigenetic mechanisms. So again, there's some examples of genetic variation. There's a lot of genetic variation. There are examples of adaptive variation. How do you decide whether there's a lot of genetic variation or little genetic variation? You decide it empirically by looking at sequence differences, and actually, compared to other primate species, genetic variation among humans is minuscule, which is an interesting empirical thing and is not what any evolutionary biologist would have expected in like 1900 if you'd asked evolutionary biologists, how much genetic variation do you think there is among humans? Well, in 1900 they didn't really know what genes were. They were discovered by Mendel and hadn't really, but in even 1940 they would have said, oh, a ton. But actually, compared to chimpanzees, so this is a tree that you should be able to interpret. You'll see trees, phylogenetic trees are not always presented sort of vertically or horizontally from a single ancestor branched out. They often are branching from sort of a center. There's a common ancestor that could go off of this branch. However, this kind of phylogenetic tree has an important character, which is that the length of the branches is proportional to sequence difference. And if you look at this tree, all of anatomically modern humans is here. The sum of all the lengths of differences of anatomically modern humans that have been sequenced is here, compared to all of chimpanzees, which is this branch, or chimpanzees or humans versus gorillas, which any of these lengths is much longer, is that length, whereas the difference among any humans are these lengths. Which is really interesting because the population size of all chimpanzees now is, I think, a couple 100,000, whereas the population size of humans is whatever it is, 7.8 billion. So why is it that 7.8 billion humans contains much, much less genetic variation than 200,000 chimps? Yeah.
Because humans are able to create oats or portable food, we require
No. I mean, humans are different than other primates in our cultural learning and tool use and a whole bunch of interesting things. But it's actually like just an empirical, weird historical fact that we discovered based on this lack of genetic variation. Yeah. Migration is part of it. Migration, yeah?
Duration since speciation. Like
The time since speciation. So some of it is the time. So anatomically modern humans have been around for about 100 to 200,000 years. You usually read 150,000 years. Humans and chimps separated about 7 million years ago by really good, robust molecular clock as well as fossil data. It's actually something sort of related to that, but a little different. Do you remember what a population bottleneck is?
Yeah.
Yeah, what's a population bottleneck? If by accident of fate a population size is reduced temporarily, then because of small population size, a lot of genetic variation is lost due to genetic drift. And anatomically modern humans, the best estimate is that, based on a diversity of genetic data, is that our effective population size before the exodus from Africa, colonization of the rest of the world and the rest of Africa, was that our effective population size was about 12,000. And that is based on the amount of genetic variation that is now existing in all human populations. So you use molecular clocks and use that lack of genetic variation. That estimates that our population size, effective population size, is about 12,000. And chimpanzees never went through that bottleneck, which is interesting. I mean, it's like, it's not trivia, but it's an important empirical fact. It didn't have to be that way, but it turns out that our best genetic data shows that we are a lot more uniform than a lot of other species, because we happen to have undergone a genetic bottleneck 54,000 years ago. That smaller population wiped out Neanderthals, colonized the world quite recently. And practically, what does this mean? Interestingly, as you know, if you take like history courses, races get defined differently in different centuries and decades. And that's because, you can kind of call them whatever you want, right? There are no hard geographic boundaries separating races, and there are no unique alleles within races as they get defined. It's kind of interesting. Like, there are differences in the frequencies of alleles that are shared among all races. And if you take 1,000 loci and you look at the differences in proportion of alleles that are 30% in this continent and 40% in that continent, and this allele that's 50% here and 30% there, you can build a statistical model that can geographically, identify where an ancestor is. But that's not the same thing as saying that there are any unique alleles separating different races. This interesting empirical fact has some interesting consequences. One is that most of the genetic variation among human groups are found within groups rather than among. It means you can be more genetically related to an individual whose ancestors were 10,000 miles away than from someone next door. It also means that human races are not the same as biological subspecies. And actually, practically, it means that we waste a tremendous amount of effort and research funding looking for drugs that will work better on some racial groups than others, rather than looking for drugs that will work better on some individual genotypes than others. Right? So there's a huge industry in racial pharmacology, trying to tailor-make drugs to different racial groups. When it comes down to it, we're gonna end up, killing or not saving groups if we maintain that, rather than targeting them to specific genotypes. Okay, that's all I got. I will see you next week. Take care. Okay, yeah. No, I'm going to be posting a study question